Good morning, and thank you, society members and the Sjöberg family, for this honor. I'm going to take a little bit of a risk here after my disclosures and try my hand at a little bit of Swedish. Jag är verkligen hedrad av att få detta pris. So. Thank you. So the rest is hopefully more understandable from here on out. So in the spirit of thanks, conventionally we thank our family toward the end, but in this case I really wanted to start off my presentation with thanks because it's not without support from my husband, Nir HaKohen, who's also in the audience here, that we've been able to build a wonderful life together, but also to conspire and dream together about how we could harness tumor immunity in the service of human health. My continuous support team have been my parents, Ching-Hui and Zita Wu, and my mother-in-law, Miriam HaKohen, and altogether we've have taken a journey to raise our 3 kids, Celia, who's in the audience, Kira and Leo, across the years, during the same years that we've kind of seen through our project in neoantigen vaccines from inception to reality. So. With that, thanks. I'd like to proceed and let's get started. So what's the challenge that we face as clinicians and as scientists? How do we meet the challenge when we think about cancer care? And that really, in my mind, is really the challenge of tumor heterogeneity. By now we know that this very fact of tumor cells is what fuels tumor evolution and therapeutic resistance. And really the successes of genome sequencing that has brought in high granularity and clear light that tumors are really quite heterogeneous, not only from individual to individual, but even within one person, that there are many different subpopulations within that tumor mass. Tumors are not growing in isolation. They are actually coexisting with the host immune system. And in this scheme, we can see that when effective, our host immune system can eliminate and even become equilibrate together with cancer cells, but all too often, by the time we diagnose our patients, escape has already happened. The tumor has evolved through many different mechanisms to either directly or indirectly inactivate the immune system that can hold the cancer in check. And this kind of idea of elimination, equilibrium, escape was well articulated by my senior colleague, Bob Schreiber and colleagues, and really has kind of led the conception of how we think that the host and the tumor cells are moving together. Now, this kind of concept, this conundrum, was appreciated early on, even in the 70s, at the first moment that we realized that cancer was essentially a disease of tumor mutations. So in this work by Peter Noel, he stated that one may ultimately have to consider each advanced malignancy as an individual therapeutic problem. In this situation, immunotherapy becomes the leading candidate for the easiest means of destroying the remainder of the neoplastic clone. And so, fast forward to where we are now, we now recognize that immunotherapy has emerged as a new pillar of cancer care, in addition to chemotherapy, surgery, and radiation. There are many different cell populations that are part of our adaptive immune response, but front and center are T cells that possess so many different type of functionalities and capabilities. And I think understanding all those capabilities leads us to understand all the different types of immunotherapeutic modalities that have emerged that were already mentioned during the introduction. But for the purpose of today's talk, I'm going to talk about vaccines, for the very reason that vaccines have really the unique ability to not only expand pre-existing anti-tumor cells that might be in the microenvironment or the draining lymph node here shown in the orange color, but also vaccines can generate new immune responses. So these are the de novo anti-tumor cells shown here in the green. And in doing so, it has the possibility of affecting tumor kill, promoting immunosurveillance, and immunologic memory. Now, so the concept of vaccine is not a new one. We are all quite aware of vaccines, prophylactic vaccines that for infectious pathogens. So we've all gone through vaccines against childhood illnesses, and in more recent memory against COVID. In the situation of infectious disease targeting vaccines, we have a clear foreign invader. There's a clear target to go after that can be quite immunogenic by virtue of being foreign, and we can quite effectively generate vaccines. The situation with cancer is quite different and more fraught. Here, as I said before, we're often dealing with patients who already have cancer, so it's not a prophylactic vaccine, but rather a therapeutic vaccine. What we're trying to do is to mount a tumor, a T-cell response against the tumor cells. In the situation of cancer, there is chronic exposure to antigens. So not only are we confronted with the challenge of genomic heterogeneity that's interpatient as well as intrapatient, but there's those mechanisms of immune evasion that I mentioned before, chronic inflammation due to the chronic antigen exposure, and the poor T cells are quite exhausted. And so, as alluded to before, we've really gone through a roller coaster of a time with generating effective cancer vaccines, but I hope we're coming out of that situation, as I'll describe. So how did I get into all of this? And really, I think this is a moment to reflect. This was alluded to a little bit by our in the introductory remarks. It was not always that those who worked in tumor immunology were the cool kids on the block. In fact, really quite to the contrary. And I think during the time that I was training as a postdoctoral fellow, there were even articles like this that we read written by luminaries in the field. This was a report of the experience in the NCI treating hundreds of patients with cancer vaccines with a tangible response rate of 2.6%. So really quite depressing for someone at the beginning of her career and thinking about whether or not to pursue this. But I do want to point out that this was some time ago, and this was the majority of those studies were really aimed at single antigens. So already what I mentioned before, tumor heterogeneity, if we understand this very fact, this was already quite a difficult bar to overcome. I am an optimist, so if you read closely in the text, there were some glimmers of hope. So there was some signal with more complex vaccines, and certainly this was something that I and others paid attention to. Now, in my case, at that time, if you were interested in tumor immunology, you either went in the direction of melanoma or stem cell transplant, because this is where we could actually see human immunity in action. And so for me, as I started my clinical training, I became very inspired by graft versus leukemia and the precedent set by a former Nobel laureate, Donald Thomas. About a number of years ago, together with my trainees, we had the opportunity to reflect on the 30 years commemoration of his Nobel Award. And just to remind everyone, what happens in a stem cell transplant is that there are normal donor immune cells that are given to patients with blood malignancies, and it's this engraftment and immune reconstitution of normal donor immune cells that have the potential to set up residence in the marrow, to begin to see the recipient cells, especially the recipient leukemia cells, as should not be there, and eliminate them, that leads to eventual cure. And I was really excited about the notion of cure and really trying to understand what was the biology behind this, like so many others. When we had the chance to write this article, we had the opportunity to reflect that really stem cell transplant has been the forebear of modern cancer immunotherapy, and many of the concepts that we confront in stem cell transplant are still the focus of topics that we try to address in modern therapies. So the notion that anti-tumor T cells are front and center of the effort, that it's important to think about the immunotherapy targets, that we have to be concerned about immune overactivation, and that combination therapy can really be quite effective. For my program, was always interested in thinking about the sharp point of the spear. So what are those donor immune cells seeing? What are those targets that we can use to stimulate the immune system? And so my program has always been about antigen discovery, and that has a direct link with how we got to neoantigens. Now, neoantigens as immune targets, it's something that we talk about a lot now, but if we think back to the early 2010s, it was really a convergence of transformative technologies that got us there. And really, if you think back to that time, there was this exciting new availability of next-generation sequencing. There was a maturation of epitope selection programs built on neural network programs that was available that would allow us to, out of a haystack, pinpoint needles that we could use for immune targeting. And as was alluded by the introducers, there was suddenly the clinical availability of immune checkpoint blockade that was forged by luminaries in the field like Jim Allison and Hanjo. And for me, for those of you who are Star Wars aficionados, I kind of akin that to like the Obi-Wan Kenobi moment when the Raider shield comes down, and suddenly we have the ability to start to penetrate the Death Star. So this is an exciting time to think about how all of this could come together. Now, speaking of next-generation sequencing, this has been a large global collective effort to understand the inner workings of cancer cells. And in the same way that next-generation sequencing has yielded to us understanding of all the different mutations and processes that are part of the cancer circuitry, we also realized that identification of those DNA mutations would provide us with a therapeutic opportunity, because certainly at least some of those mutations would have the ability to be transcribed and translated into peptides that would be processed, and at least a portion of them would have the ability to become presented in the context of HLA alleles on the surface of tumor cells where they could be recognized by T cells. So this concept of neoantigens, these amino acid changes rendered by the DNA mutation that would create immunogenic peptides, this was not a new idea. It had long been conceptualized that tumor neoantigens would be hitting the sweet spot, really be the optimal tumor antigen to go after, because by virtue of being born of those tumor mutations, they are extremely exquisitely selective for expression within the tumor cell, not normal, and then akin to pathogens, highly immunogenic because these were somatic events, and that there was a lack of central tolerance against them. And this was really different than where we had been in previous generations of studies to target and create cancer vaccines that were really concerned with shared overexpressed tumor self-antigens that were variable in their tumor-specific expression and variable in their degree of immunogenicity. Although this was kind of conceptualized as the optimal tumor antigen, recognizing that the panoply of neoantigens would differ from individual to individual, it was always a conundrum how one could feasibly go about identifying neoantigens on a per individual basis. Well, next-generation sequencing solved that problem because suddenly we had a feasible approach that would allow us for any tumor cell to go through that sequencing process, identify those mutations, and with knowledge of the HLA type of that tumor cell, we could then also use prediction to generate for us a list of candidate neoantigens that we could go after. And so this was really the basis of the work that we took forward. From my perspective as a stem cell transplanter, it was not a huge leap of faith because we already had several lines of evidence in our graft versus leukemia work to support that this would be really a feasible strategy. In my own group, we had already performed whole tumor cell vaccines following stem cell transplant, and if you follow the red line on the bottom, we could already see that we could detect immune responses that were tumor specific for individuals. This was also the age where there was many targeted kinase inhibitors that were becoming available. So among the first was Gleevec, imatinib for chronic myelogenous leukemia, formerly an area of active work in the area of stem cell transplant. We knew even within the first few years after FDA approval of Gleevec that mutated BCR-ABL was emerging as a resistance mechanism. And the question that we asked in our group was, could any of those mutated BCR-ABL mutations generate epitopes that would be immunogenic? And some early work that I did together with a very talented medical student, we in fact could generate tetramers against a A3 restricted peptide and identify that in a stem cell transplant patient who actually had this allele and this mutation who experienced remission, we could actually identify a T cell population circulating against this mutated epitope and see that there was immunogenic responses that were evident following transplant that were not there before. And finally, the very fact that genomic changes could generate an immune response. Well, we already had in stem cell transplant evidence of that. So those of you who know graft versus host disease, this is immunologic sequelae of immune recognition of minor histocompatibility antigens. These are antigens that arise from single nucleotide polymorphisms that differ between donor and host. And we already understood by the time that next generation sequencing was available to us that such immune responses could generate many different sequelae across different target tissue depending on their expression of these molecular differences. So it was truly exciting over the ensuing number of years to follow the work that was conducted worldwide to look and poke holes and see if neoantigens could be effective tumor rejection antigens. In general, there were 3 buckets of evidence. One bucket was to look at the concept of neoantigen load. So if neoantigens were really immunogenic, shouldn't it be that tumors that have a higher load of neoantigens would have better clinical outcome? And in fact, across studies in TCGA or in specific tumor settings or even following immune checkpoint blockade, in fact, this was consistently a type of result that was seen across many groups. Another bucket of evidence was to ask, well, if neoantigen-specific T cells are participating in effective anti-tumor immunity, cannot we look at settings where there is effective immunity that's clinically detectable, and shouldn't we be able to see evidence of expansion of those T cells that are neoantigen specific? And our group looked in the setting of stem cell transplant, but certainly other groups looked in the setting of TIL therapy and immune checkpoint blockade. And finally, if neoantigen-specific CTLs are directed against an antigen that is a bona fide tumor rejection antigen, should we not see evidence of in vitro and in vivo tumor kill? And certainly there was very beautiful studies, both mouse and man, in the groups of Bob Schreiber, Tyler Jacks, also human studies by Steve Rosenberg and Eric Tran that could directly demonstrate that infusion of T cells that were neoantigen specific would lead to decrement in tumor burden. And so really all of this evidence together as a community has led to a paradigm shift that we can conceive of thinking about a multi-pronged attack focused on neoantigens that is directed specifically against the tumor that would not affect normal tissue as collateral damage, and really thinking about high immunogenicity and tumor specificity. And this has kind of led to our conception of thinking about cancer vaccines as a way to steer the immune response, that if we have a way to release the brakes and we have a way to accelerate the response with good adjuvants, can we not use cancer vaccines as a way to expand and broaden the T cell repertoire by inducing tumor-specific T cells that are highly specific for the tumor cells, but with minimal damage and side effects to normal vital tissues. Now, our question, having kind of generated this framework, was can neoantigen targeting vaccines actually stimulate tumor immunity in our human patients? And in my first R01 as a starting out investigator, I kind of laid out kind of a roadmap for how we would try to examine the functionality of T cells that had neoantigen specificity. So this is reviews from that first application, and certainly there was reservation about the translatability of the work, considering the sophisticated processes of identification and characterization. And in the end, the reviewers were moderately enthusiastic. And so I am an optimist, but my family will also tell you that I have a little bit of a contrarian streak in me. So this type of tepid enthusiasm probably fueled our determination even more to kind of pursue this path. So we early on came up with this concept of how we would go from tumor biopsy to treatment of patients. And it was really the idea that could we not in real time procure tumors, perform DNA RNA sequencing to find those tumor-specific mutations, together with HLA typing, as I mentioned before, perform prediction on the personalized HLA binding peptides, and come up with a manufacture strategy that we could use to generate the vaccine. For the time that we, at the time that we kind of conceived of this, we decided to go the direction of synthetic long peptides. So not the 8, 9, 10 mers when we're thinking about class 1 restricted peptides, but rather 20 to 30 mers, because there had already been beautiful published work by Kees Malief in the Netherlands to demonstrate that these longer peptides would require internalization by professional antigen presenting cells, and in doing so mobilize not only a CD8 response but also a coordinated CD4 response that would probably be needed to generate a robust immune response. We also decided to go after multiple targets, thinking about the tumor heterogeneity, so up to 20 targets that we wanted to put together with a strong adjuvant, in this case poly-ICLC. And then we were also thinking about how to put this together with immune checkpoint blockade, and in total get this into a syringe to give to our patients. And not to go over a lot of published work, but our studies are in the red. We were able to co-publish our studies together with Ugur Sahin of BioNTech that reported out our responses in high-risk melanoma. This was 2 years after the work of Beatrice Carreno and Jerry Lynette. All of us used a computational genomics-based approach to find neoantigens, and Carreno et al. Used this to target dendritic cells, whereas Ugur Sahin used an mRNA-based vaccine. Subsequently, we also looked at newly diagnosed glioblastoma, and our work was published concurrently with groups in Germany who also used this type of genomics computational-based approach for personalized vaccine. And I would say that in total, all our studies together have demonstrated the safety and feasibility of this approach, that it's highly immunogenic, and in fact is promising in combination with immune checkpoint blockade. So I'll show you in the next couple slides some of the results. Probably some of our most exciting results from our first study came from the example of these two patients. So these two patients had stage 4 melanoma, so really quite advanced disease. In the blue, you could see that they received neoantigen vaccine, and it was not surprising that they both suffered a recurrence thereafter. That's shown in the red triangles. But what was surprising is after their treating physician started them on anti-PD-1 antibody, shown here in the yellow, both of them within 12 weeks achieved a complete clinical response, which is highly unusual for patients with this type of melanoma. And I'm happy to say that both patients now, years and years later, remain alive and doing well off all medications. This is just some of their scans from the time that they started anti-PD-1 therapy. You can see evidence of disease that melted away within 12 weeks after initiation of anti-PD-1 therapy. So this was, if you think back to the cartoon that I showed before, combining cancer vaccines with immune checkpoint blockade, this was certainly a promising evidence that we were moving in the right direction. I'm going to take you through 4 other different types of results that we've seen over the years in monitoring our patients that have led us to conclude that there are encouraging short and long-term responses to our vaccine. So one conclusion is that we do see generation of a memory response. In those melanoma patients, we were able to generate tetramers to a number of different neoantigens from a few different patients, and from those neoantigen-specific T cells that we pulled out from the periphery, we performed single-cell transcriptome sequencing, and we were able to define different clusters of T cells, which when we started to parse out and look at these cells in a time-resolved fashion, we were able to actually see that there was a cascade of transcriptional profiles, initially mobilization of a cytotoxic response, and then contraction, and ultimately generation of immunologic memory, which is exactly what we hope for when we give patients their vaccines. Secondly, from those individual single cells for which we looked at the transcriptional program, we were also able to gain TCR, T cell receptor information, and this allows us to look at the T cell clones over time. And so what we saw here, if you follow these pie charts where each color denotes the first time that we see a T cell clone, you see that there's red that persists across time through the priming and the boosting phase of the vaccine, but you also see increasing colors as you shift from left to right, demonstrating that there's actually a diversification of clones that are neoantigen specific over time. Functionally, and this is an ELISpot assay, so this is an assay that we use to where there's interferon gamma secretion that we see with each of these individual dots in these wells here in triplicate. We see that there is as strong immune responses against those mutated peptides that were part of the original vaccine week 16, but also close to even 4 years later. So there is long-term protective responses, circulating responses that we can detect from our patients following vaccine. Where are those T cells going? We were also, because of the TCR information, able to track that the neoantigen-specific T cells were tracking to the site of tumor. So I already talked to you about those patients who generated fantastic responses after vaccine and immune checkpoint blockade. This is one of those two patients where you see we were able to draw blood at the following priming and boosting of the vaccine, but also at the time that the patient was on anti-PD-1 therapy. And the overlap between the green and the orange tells you that there is a persistence of dominant clonotypes over time. In the red are those clonotypes that we know are neoantigen specific due to our single cell studies. And what's super interesting is that because we had these molecular barcodes, we could go into the patient tumor at the time of recurrence, and through bulk TCR sequencing, we could see that actually there was a great deal of matching of those molecular barcodes, indicating that those neoantigen-specific T cells were actually able to go to the site of recurrence. And one may speculate that perhaps the reason why there was such a rapid response once exposed to pembrolizumab is that those soldiers of the army were already on site, ready to be mobilized once the anti-PD-1 hit. We've been able to see this tracking of neoantigen-specific T cells at the site of disease in a number of other systems. So in another study where we looked at glioblastoma, here we had performed immunohistochemistry over time following vaccination at the site of recurrence in GBM, and we had already seen that there was increased CD4 and CD8 infiltration. I want to say that glioblastoma is as many of us know, is a super tough disease, one where there really is inadequate T cells that are typically at the tumor bed. But again, with tetramer-assisted isolation of neoantigens of T cells, we were able to perform single-cell sequencing. Here, each circle is a T cell. If they're together, that means they are of the same clonotype. They were either, from single cell, we could see that they were either CD8 in the orange or CD4. What was super interesting is that if we took T cells from the peripheral blood, stimulated against with the neoantigen, and performed TCR sequencing so we knew exactly those clonotypes that were related to the neoantigen, we saw that there was overlap between those T cells and what was directly infiltrating the glioblastoma. And when we took those overlap TCRs and cloned them, we could confirm that, in fact, those TCRs were directed against the mutant peptide in the red, not wild type, that was part of the vaccine mixture to begin with. So this, again, told us that what we had given the patient as an injection in the skin mobilized T cells that were actually able to track to the tumor, so getting soldiers on site. Recently, in work performed by Vinod Balachandran and Memorial Sloan Kettering, we've also seen evidence that this type of phenomena can also happen in pancreatic cancer when those patients are given an mRNA-based vaccine. So in there, again, a really super tough disease to address. But in their study, about 50% of patients were able to generate an immune response, and those were the patients who did very, very well, so lack of any progression over the course of several months. And here, what they also identified, there was one patient where they suspected that there was a recurrence, and there was a new liver lesion, but then after vaccination disappeared. And when they did a deep dive of this particular individual, they could see from this lesion that there was no visible tumor, but there were many, many T cells. And as they characterized those T cells based on their that molecular barcode, the clonotype expression, they could see that these were all vaccine-expanded T cell clones. Moreover, when they did molecular typing of the tissue, they could see the presence of a mutation referable to the original tumor. So again, evidence that the tumor had been there, supporting the idea that there was immunosurveillance and actually elimination of tumor cells. So a final piece of evidence that we've seen in terms of our results following vaccination is the phenomena of epitope spreading. So this is the notion that although there may be an initial tumor kill from the vaccine, so if you follow the blue as the antigen, that by virtue of release of tumor contents, there may be a secondary wave of immunity, so that over time there's not only immune responses against the vaccine antigen, but also the non-vaccine antigen. And we've been able to see this in our melanoma study. So here, this is after priming and boosting, we can see a new specificity against a new antigen that was not part of our original vaccine, and this persists even weeks and weeks later. And in a larger study performed by Neon Therapeutics, where they also looked at the concept of epitope spread in patients with melanoma, they saw better clinical results in those that had epitope spread versus lack of epitope spread. And so it's been a long road, but I kind of wanted to lead you through the different steps and paths that have led us to consider these encouraging results. As a community, we've seen that this is true whether the vaccines are coming from synthetic long peptide vaccines or mRNA vaccines. And what's been super exciting is that over this past year, we've seen the first randomized phase 2 trial that has demonstrated clinical benefit to personalized cancer vaccines in the setting of melanoma. And these are the results that Jeff Weber presented last year in AACR and now in Lancet. And it shows that there's a benefit when the mRNA vaccine here produced by Moderna was given together with pembrolizumab compared to in the blue pembrolizumab alone. And just, it's, again, super exciting because in the next couple years, I think we're going to see results of larger scale mRNA vaccine studies supported by industry, whether from Merck, Moderna, or Genentech, again, with the concept of using multiple different neoantigen targets across different disease settings, enrolling hundreds to even thousands of patients. So for someone who's really been kind of from the beginning involved in studies of just a few patients, it's really exciting to see this kind of being taken to the large scale. Now, Is there more to do? For sure there is more to do, and even as we recognize that we've been really pleased that our work has been recognized, we realize that there is a great need to continue to do deep dives into trying to understand tumor immunology, the tumor aspect, as well as how to harness tumor immunology. This is a figure that I generated together with our colleague Ed Fritsch, who really loves San Francisco and the Golden Gate Bridge. So this was kind of an image that he pulled together, and we talked about the troubled waters in San Francisco Bay that cancers generate, but also the pillars of pulling together vaccines that we need to pay attention to. Who are the cellular players? What is the antigen? How are we going to deliver? What are the co-therapies that we have to bring together? And so the way that my group has really been focused and thinking about how to work on this is to continue to go with the paradigm of bedside to bench and back again efforts. So through the lens of thinking about clinical trials and how we can tackle different questions to improve our vaccine outcomes. By virtue of our clinical trials, we have really dedicated biospecimen collection, and this affords us opportunities to look at disease-specific research, and we are always looking for opportunities to maximize what we can learn from our human biospecimens. And so I'll take you through a little bit of the yellow, orange, and green zones of our work. So really briefly, in terms of our ongoing trials, I've really had the good fortune to work with really terrific clinical investigators at our institute, and by now we have a portfolio of different tumor types that we've been going after, each one designed to ask a specific question and to test specific modalities and co-therapies. I just want to share some recent results, still unpublished, but that we're super excited about. This was a study led by David Braun, who's now a faculty member at Yale New Haven, and he really oversaw a study in kidney cancer where we took patients with advanced disease and following total resection, provided them with a vaccine that we administered in a prime boost schedule together with subcutaneous ipilimumab. And what we've seen is super promising results. So in this adjuvant study, even though kidney cancer is a cancer with a low mutation burden, we were still able to identify multiple targets that we could use to construct the vaccine, and we've seen really fantastic results even 40 months from the time of first vaccination. The immune responses have been super robust, including cancer drivers. So the von Hippel-Lindau gene here mutated. We saw super strong immune responses, and we've looked at a number of other RCC drivers. If you follow the red line over the course of months to years, we've seen persistence of vaccine-associated immune responses over time. And finally, we've been able to show that these responses can be detected against the autologous tumor itself. And so, of course, this is what we want to go after. Now, in terms of our disease-specific, taking us to the orange zone, we wanted to take a deeper dive into the tumor microenvironment because we understand that it is quite a hostile environment in which we are sending our vaccine-induced T cells. We also understand that in the native tumor microenvironment, there's a relative paucity of tumor-specific T cells. There's a lot of bystander cells. So how can we actually get to the cells that we want? And this has really motivated a lot of the single-cell work that my team has performed, and we've been really interested in the transcriptional profiles of the immune populations coming from cancer specimens. We've wanted to link that with the architecture of T-cell immune responses, and we've also wanted to know, again, what are those T-cells seeing? So what is the antigen specificity of those T-cells that are tumor-specific? And so in the realm of kind of tumor profiling, we've, for example, David Braun took a deep dive into RCC, and he's been able to identify a circuit of exhausted T cells together with M2-like macrophages that seems to be really quite operative in this cancer setting. In another setting, we've looked in chronic lymphocytic leukemia, and we've appreciated that even when we look at what would be considered a pre-malignant state, so monoclonal B-cell lymphocytosis, those alterations in the composition of immune cells are detectable even very early on, before frank malignancy, telling us this immune activation is really a conundrum that we have to address, that is so much a part of the cancer picture, and so really quite a challenge to meet. So in terms of trying to get to understanding the antigen specificity of those T cells, we've had to develop new approaches to address that challenge. So if we're able to identify expanded T cell clonotypes, can we rapidly clone them and basically at a single cell level? In this case, what we've come up with is a parallelized miniaturized strategy using Gibson assembly to clone the CDR3 alpha beta sequences that define the TCR. And then we have come up with a combinatorial dye labeling strategy for once we introduce the TCR into our vehicle T cells, so that in one tube we're able to actually, with these different dye combinations, we can feasibly interrogate them with different formats of antigen, and through gating strategies, look at them in parallel. So through the strategy, we can either look at reactivity against tumor or reactivity against specific antigens that we may identify either through the mass spec or from prediction or even existing known pools of antigen. And this is just an example of some of the readout that we see by flow cytometry. We're using 41BB as a readout, and in this example of a tumor-specific TCR, we see reactivity against melanoma or interferon gamma-stimulated melanoma, but not against autologous PBMCs or B cells or even EBV-immortalized B cells. So this is just to give a flavor of some of the work that we've been conducting, but in this example of melanoma, we've been able to transcriptionally evaluate the T cells within the melanoma microenvironment. If you just focus on what we would call the red colors, so the exhausted T cells, the phenotype is that of an exhausted T cell. In the blue are what look like a non-exhausted memory T cells. And when we started to clone out expanded TCRs that were either from this red zone or the blue zone, and test them against a tumor, we realized that it was really in this shape, which we call a turtle. It was in the head of the turtle that we saw that all the tumor-specific TCRs were residing, whereas the viral specificities seemed to be in this blue zone. And from this work, we've been able to generate putative tumor-specific CD8 signatures, as well as viral-specific signatures. I won't go into it today, but we've been since able to look in other solid tumors, such as head and neck cancer and kidney cancer, and we see this sort of alignment of tumor specificity, exhausted phenotype, seeming to kind of recapitulate itself across different settings. In the melanoma case, as we start to look at antigen specificity, we begin to see that across the 4 patients for which we did a deep dive, looking at hundreds of TCRs per patient, we could actually see that there was really a different profile antigen by antigen across the patients. So here are the targets that are melanoma associated. In the blues and greens and purples are the neoantigens, and you can see that patient A and B is really melanoma associated antigen dominant, whereas C and D, you see more neoantigen profiles. And this is beginning to raise up the question whether there's tumor intrinsic features that are driving the antigen specificity landscape that we see. And in future work, we're trying to understand how this then relates to the type of responses that we see against vaccine. Again, thinking about the T-cell clonotype as kind of a center of some of our activities, we've also been interested, as we move into the green zone, thinking about how we can look at those neoantigen-specific T-cells in space. And so we've really begun to take a deep dive into spatial transcriptomics using the SLIDE-seq technology. We work together with Fei Chen to develop SLIDE TCR-seq, so this allows us to look at the transcriptional profiles in space, not only of the various immune cells, but also to single out, again, by virtue of amplification of their TCR receptor CDR3 alpha beta sequences, and localize them, and we can begin to ask questions such as, what is the spatial localization of exhausted versus non-exhausted memory T cells? What about those neoantigen-specific T cell clones within the tumor? What is the crowd that these T cells run with? So what are the neighborhoods that we see? And so just to kind of give you a sense of the type of studies that we're doing, again, in the melanoma setting, in this tissue where we were able to isolate different regions of interest, we were able to see in this particular specimen that there were really two different lobes that we were looking at, in the blue and in the orange. But what was super interesting when we started to superimpose our TCR-seq workflow is that we saw that there was one particular T cell clone that was enriched in one lobe, the orange lobe, but not in the blue. And so as we started to look at the neighborhood around that T cell in terms of neighboring macrophage monocytes, we were able to see that there was enrichment of CXCL10, which we know is a T cell attractant. And then when we looked at that T cell in relationship to tumor cells, we saw that there was a downregulation of MGST1, which has been implicated in cancer immunotherapy response. And so I think that this is kind of the starting point of some really exciting studies that are currently ongoing. So just to summarize this part of the talk, I would say that personal antigen targeting addresses tumor heterogeneity. And our current series of clinical and translational results are certainly encouraging. We have been fortunate to be able to generate investigator-initiated clinical trials, and we really have strong cooperation of motivated patients that have been very generous in giving us time and going in for the extra blood draw that has allowed us to monitor their immune responses over time. I think that understanding the tumor microenvironment is one of the foremost challenges that we have to unlock. At some level, when we start to look at the tumor microenvironment, I kind of think about this a little bit as archaeology. We see past hallmarks of the 3 E's. We can see evidence of T cells that kind of came, recognized, and were inactivated by exhaustion, but for sure have clear specificity against the tumor cells. We do see similarities and yet stark differences across the diverse tumor types, so always needing to customize what we're doing on a per cancer setting. And we do have major opportunities for novel tumor antigen discovery and prediction, as well as novel delivery approaches. And so I would say that for the current time, our neoantigens that we're going after are really in the realm of single nucleotide missense mutations or insertion deletions, but there are many, many other classes of genomic alterations that may be more prevalent in certain tumor types, maybe what we deem the low mutation burden tumors, that I think we hope will be fruitful in future studies and are potentially targetable. I think just to close, I just want to say a few words about our vision for the future and our goal for getting to a cure. I've had the good fortune of working with a lot of trainees, and certainly some of our work we hope inspires young minds to kind of go into this very active field. It's been really delightful for me to have a correspondence with a young budding scientist who is the daughter of a colleague, Eleanor. And she wrote to me early on in the middle of the pandemic, and she said, "Kathy, I think I have a cure for cancer." And she really kind of was trying to give me some advice, and in the end she said, "I also think that you should put it in through a shot." So love, Eleanor. So this is a correspondence that we continue to have, and trying to think through all the different aspects. But I think, I think taking a step back and thinking about the lessons we've learned so far, I think cancer vaccines can really, I think, are a very important therapeutic modality. I think it can really fit in all aspects of the cancer treatment schema, thinking, we'll go from right to left. So when we think about advanced disease, how we can try to eliminate tumor at the time of advanced or metastatic disease, we can think about it from the perspective of prevention of relapse after first diagnosis, but ultimately we want to get to cancer prevention. And just thinking about these three different points, I think first for advanced disease, I think what we've learned is that cancer vaccines are a little bit more subtle than, for example, cellular therapy or even immune checkpoint blockade. But I think it works quite effectively in combinations. So we do need to think about what are the right combinations that we do need to treat this very thorny problem. I think the greatest successes we've seen so far clinically have been in the prevention of relapse. So, and I think that speaks to the fact that the immune system is always going to work better in settings of minimal residual disease. And as I showed you before in the kidney cancer case, that is a setting where there's total resection, and then we can help along with immunosurveillance and generation of memory. But ultimately, I think our biggest wins are going to be if we can get to early interception, because of the potential for long-term impact in morbidity and mortality. But what is really required then is to consider cost and time for manufacture, because we really need to get to large populations of patients safely to be able to see and make a difference. And again, my great thanks to the Sjöberg family for the prize, and we will put the prize money to good use and work on these problems. So really deep gratefulness and appreciation there. I think as I kind of think forward to the next couple years, my hope is that with all our knowledge about cancer genomics, we can tailor our vaccines to the specific mutations of patients, but I also do hope that we can also integrate immune profiling. So if we knew whether a cancer, for example, was immune inflamed or immune excluded or immune desert, could we not figure out how to sequence or provide those combinations in sequence that made sense for that individual patient? And here is my last slide to say that I do hope in the not too distant future, patients can go to their doctors, review their genomic results or their immune profiling results, and be able to call directly to the pharmacy and say, let's order up a personal cancer vaccine. And the pharmacy says, yep, it'll be ready for you in a couple weeks, come back. So that's the dream and that's the hope. I really want to thank my funding sources for the work, and many, many thanks to the Sjöberg family for providing me with this really extraordinary opportunity. But I think that I also want to deeply acknowledge and the privilege I've had to be working with extraordinary individuals. These three people has really been the brain trust that we've had for our project. So Nir HaKohen in the audience, Ed Fritsch, and Patrick Ott, clinical investigator at Dana-Farber. But all these studies, I talked a little bit about the origins about our work, and so I'm deeply grateful to my senior colleagues at the Brigham Women's Hospital and in hematology that gave me my early taste of what cancer immunotherapy via transplant could mean. My transplant colleagues, Rob Seufert, Ted Allier, my mentors in immunology, Alice Reinhertz, Glenn Dranoff, and Gordon Freeman. And most importantly, in the early part of my career, and even now, my mentor, Jerry Ritz, who has really taught me so much. I've also had the good fortune to work closely with colleagues at the Broad Institute and had an early vote of confidence from Eric Lander of the power of genomics and making an impact on medicine. And I've had the privilege to work with so many other extraordinary people across the platforms there. across the Harvard Medical School community, and then I mentioned the clinical investigators, but there's really been really wonderful community of biostatisticians, immunologists, computational folks that have made these vaccines happen, and so many trainees that have brought their energy and their excitement into the work. And I'm sure that I'm missing people, so and I deeply apologize for that, but it's really been a village to make all of this work happen. So thank you for your attention, and thank you.